Hello class, Dr. Barr here. Today we are going to talk about Chapter 1, which covers the main themes of microbiology. This chapter from your Foundations in Microbiology textbook is going to briefly cover the topics that we plan to cover in a great detail throughout the semester. But first, my question for you is, what is microbiology? I have had students in the past tell me that microbiology is the class that constantly reminds you that the worst horrors are invisible. Although some students may believe that to be true, the real definition of microbiology is the study of microorganisms. Microorganisms are extremely small, but they do vary in what we call a small size. Anything that we term a microscopic organism, that means it's a microorganism or a microbe, something that is too small to see with the naked eye. We need some sort of assistance to see these organisms, whether that be a microscope or some other sort of technology. The major groups of microorganisms that are studied in microbiology and that we will cover in this course include bacteria, viruses, fungi, protozoa, algae, and helminths. Microbes are what we call ubiquitous. This means that we can find them in pretty much all natural habitats. So even in environments that we find to be too harsh, such as in Antarctica, where it can get to be below freezing, very below freezing, or even in the hot springs of Yellowstone Park, where some of the hot springs can become extremely boiling temperatures or even high acidity, we do find microorganisms living in both of those extreme environments. Before going any further, I'd like to quickly talk a little bit about the history of microbiology. The science of microbiology is credited to be born in around 1674, thanks to two gentlemen, Antony van Leeuwenhoek and Robert Hooke. Antony was a fabric merchant who was very interested in the world around him. He created this very simple magnifying glass, as you can see here. And then he used that to study lake water. He would take samples from a nearby pond, and then he would look underneath to see what he could observe. He drew these pictures of these moving, some of them are moving, but of these small creatures that he termed animalcules. This is kind of the first credited recording of microorganisms within our environment or around us. Robert Hooke, around the same time, also is credited with discovering microbiology. His was a little different. He observed fungus or mold growing on bread that had spoiled or gone bad. He noticed that the filaments would spread out or grow, and he kind of explained or hypothesized that this mold was actually created from very small microscopic or too small to see organisms and cells. Something else that we need to talk about in terms of microbiology history is that really these concepts that we are going to talk about throughout the semester, or even concepts that you have in your daily mind and that we kind of take for granted. Centuries ago, some of this what we call common knowledge was not common. It was something that may not even have been thought of. For example, if you have ever left meat out or maybe a banana out and a few days later you see either maggots or flies sitting on the spoiled food, we wouldn't assume that it came from eggs that were hatched. So these eggs that came from other bugs, and then they hatched and they formed the bugs that you now see. To us, that's common knowledge. That's something that we know is to be true. However, centuries ago, that was not the case. Many people believed in something called spontaneous generation. And that is the idea that life arises spontaneously from non-living material. So those fruit flies that people may see on bananas, they just assume they appeared. They appeared out of nothing. The fruit flies came from nothing. They came from the banana, essentially. Same with rats that would be on ships. These people would assume that it just arose from the garbage on the ship. Now we know rats come from other rats. But like I said, centuries ago, that was not the common thought. People assumed that life could spontaneously arise from other non-living material. Once again, calling that spontaneous generation. It wasn't until 1668 that Francesco Redi was able to demonstrate using a scientific technique that worms on rotting meat actually came from eggs of flies that landed on the meat, would lay the eggs, and then those eggs would hatch and give rise to those maggots. Despite doing these experiments and suggesting that life has to come from life, people were not convinced. People were very set upon this idea of spontaneous generation. There were a lot of people who supported spontaneous generation and even did experiments that did support it. There were people who would do experiments that contradicted this idea of spontaneous generation and instead suggested that life has to come from life. So there was a lot of this back and forth, and I would say that the reason why there was a lot of back and forth was that everyone was kind of doing their own experiment. They weren't properly repeating each other's experiments. So we were getting a lot of this discrepancy or this difference with the results. This controversy continued until Louis Pasteur came along, and we kind of call him the father of modern microbiology. He did this fantastic experiment that people can easily repeat today, and we call it Pasteur's experiment. What he did is he took this swan neck glass. As you can see, it's a glass, essentially a glass sphere with this swan neck glass tubing. Within it, he placed a broth, and then he boiled at extremely high temperatures for an extremely long period of time. That boiling would would have eliminated any microbes that were within the broth, essentially making it what's called sterile. Sterile meaning that the broth is free of any sort of living organism. This curved swan neck is very important. The reason for that is because of gravity, we're going to try to have organisms falling into the glass container, but because we have this curve, they're essentially going to get stuck in this little area, and they're not going to be able to go in the broth. So if you look at this particular image on your slides, you will see that the microbes get stuck within that curved part of the neck, and they're unable to enter the broth. Therefore, the broth remains sterile when it's kept in that particular position. Pasteur was able to show that even after months after that initial boiling, the broth was still sterile. There was no living organisms within it. It wasn't until that he either tipped or broke the glass neck, allowing microbes into the flask, that's when he would actually see growth or contamination of his broth. Therefore, this evidence supported and eventually led to the formation of what we call the theory of biogenesis. The theory of biogenesis describes that all living things must arrive from other living things, or essentially life comes from life. Things do not spontaneously appear. Life forms do not spontaneously appear. They must come from another life. You're probably thinking, well, yeah, of course, Dr. Barr, that's pretty obvious. To us, that is extremely obvious. Like I said, to us, that is common knowledge. That is something that we take for granted. Centuries ago, that was groundbreaking. This was a major turning point for the field of microbiology. Another thing that Louis Pasteur did, or that came from his experiment, is this idea of having to create experiments that can be easily reproducible by others. And eventually this led to what is called the scientific method. The scientific method is made up of multiple steps. first an observation is made. In regards to Louis Pasteur, his observation was that broths could become contaminated or could have living forms. His question was, where are these microbes coming from? So next up is question, why is this observation happening? Then you want to develop a testable explanation, which we call a hypothesis. This is what you want, basically your answer to the question. At this point, it's still a guess, and that's okay. His prediction was that the microorganisms that were in his broth had to be coming from other microorganisms. Once you have a hypothesis, you want to be able to test it. Try to do some sort of experiment that's either going to support it or disprove it. It's okay if you're wrong. Science involves a lot of being wrong, and that's okay. So Louis Pasteur, for example, made all those experiments with the swan neck flasks and the boiling. You want to make the experiments in a way that you can easily do. As you're doing the experiments, you want to collect, you want to analyze data. So you want to have what we call raw data, RAW. This is just the numbers, so the physical recordings of what you're seeing. You can then take that data and put it in some sort of analyzing form, whether it be a graph or charts or images. Once you have all your data analyzed, you can take that and then draw a conclusion. Was your hypothesis correct? Maybe you found something entirely different. So for Louis Pasteur, the conclusion that he reached was that microorganisms, or those microbes that he saw within his broth, had to have come from other microbes in the air, which led to his theory of biogenesis, so the idea that life must arise from life. A very important part of the scientific method is being able to communicate your methods, results, and conclusions. You want to be able to share this information with others. It doesn't do much good just sitting there in your computer files or just in your mind. We want to share this with the world. Okay, knowledge is power. We want to be able to have other scientists be aware of your findings, and then they can take that and repeat it to help support your findings. Or maybe they can use that information and come up with even more experiments and more predictions. It's very important when these methods are communicated that they are communicated thoroughly, they're easily to read. Make sure that the methods are very clear. So you need to explain, what was in the broth, what kind of broth was it, how much broth was used. Okay, they have to be very specific, easily reproducible. So if I were to do an experiment, I need to write out the methods that I can then basically turn and hand to you. And then you can take that and do the experiment on your own. We want to make it very simplistic and easy to reproduce. You want to share your results with others and your conclusions. If enough people are able to reproduce your experiments and draw the same conclusions, that can lead to what we call a scientific theory. The scientific theory is an explanation supported by a large amount of evidence. So I can't just come up with a theory by myself. I need to do the experiments and have a lot of people do that same experiment and come up with the same results every time, or the same evidence every time. Something I want to point out about the word theory. In the English dictionary, theory means something that is proposed, or maybe something that is guessed at. Okay, it doesn't mean true necessarily. In the scientific community, however, theory is kind of what we hold as the accepted explanation for something. So in science, a scientific theory is pretty much not quite law, but it is the accepted explanation. It's the truth, essentially. So the theory of biogenesis, even though we have that word theory in front, the theory of biogenesis means life comes from life. That is well accepted. We have so much evidence that supports that, and I'm sure you would also agree that that is very true. Life comes from life. As a scientist, something that I always want to point out to others is that you can never prove a theory. You can only disprove, or you can support a theory. The reason why you can never prove a theory is because thanks to Albert Einstein, he made the wise, or however you want to explain it, he said that you can never prove a theory because it just takes that one time, that one set of evidence that can make a theory incorrect. So because you can never do every single experiment in the world to actually prove a theory, it's just not possible. So you have Albert Einstein to thank for that. Okay, moving on past the history. All of life can be broken down into what we call 3 domains. We have archaea, bacteria, and eukaryotes. And we're going to quickly talk about each 3 of these very briefly. We will cover archaea and especially bacteria in great detail when we discuss chapter 4. Eukaryotes we will discuss in great detail when we discuss chapter 5. Like I said, there are 3 domains of life: archaea, bacteria, and eukaryotes. A term that we can use to describe archaea and bacteria is something called prokaryotes. I want to make something very clear. Prokaryotes, in itself, is not a domain. It is simply a characteristic that we can use to describe both archaea and bacteria. Prokaryotes in Latin translates to pre-nucleus. Therefore, prokaryotic cells lack something that is called a nucleus. A nucleus is a membrane-bound organelle that houses the genetic information of a cell. Eukaryotic cells have this, but prokaryotic cells do not. As you can see in this image here from your textbook, this is a very simplified, general image of a prokaryotic cell. As you can see, there is no nucleus. Instead, within the cell, we have the chromosome or the genetic material just within the cytoplasm that's not contained within a nucleus. However, it is somewhat organized in that general area where we find our genetic information. We call that a nucleoid. We are going to talk a lot more about prokaryotic cell characteristics and the different parts of the cell within chapter 4. I just want to give you a quick introduction about what a prokaryotic cell. Remember, no nucleus. It is a characterizing factor. It is not a domain. The domains are archaea, bacteria, and eukaryotes. So prokaryotes means the domain bacteria is one of the 3 domains of life. Bacteria we will cover in a lot of great detail when we talk about, or as we go throughout the semester. As I mentioned, bacteria does not have a membrane-bound nucleus. That's why we call it a prokaryote. It also does not have any other membrane-bound organelles, something we'll discuss more in chapter 4 and 5. DNA is still contained within somewhat of organized fashion, and we call that a nucleoid. They do have a specific shapes that are rods, spherical or spiral. Their rigid wall contains something called peptidoglycan. This is unique to bacteria. We will only see this within the domain of bacteria. Many move using what's called a flagella. Archaea is another one of the domains of life. Archaea are prokaryotes. They're similar in shape, size, and appearance to bacteria. They multiply via what is called binary fission. That means that one cell, we begin with one cell. That cell is going to equally split into 2 daughter cells, meaning those 2 daughter cells will be identical to that parent cell. It's the same process that we see in the domain of bacteria as well. Archaea cells have rigid walls, however, they do not have peptidoglycan. So it's a very major distinguishing factor between bacteria and archaea. Bacteria have peptidoglycan, while archaea do not. There are also ribosomal RNA sequence differences. We'll talk about a lot of that in chapter 4. And then many are what are called extremophiles, meaning that they like extreme environments. They like either high salt concentration, extremely high temperatures, or extremely low temperatures. Once again, we'll discuss a lot more of this in chapter 4. Domain eukaryote, we will talk a lot about eukaryote cells in chapter 5. Eukaryotes, that in Latin translates to the true nucleus. So they have this membrane-bound organelle within their cells. It contains the nucleic acids, so the genetic information of the cell. There are also other membrane-bound organelles that we find within the cell. These are definitely more complex than prokaryotic cells. When we talk about different organisms, we sometimes may give it a general name or a common name, but all living organisms also have what we call a scientific name. It is very important to know how to properly write out a scientific name. Whenever you write out a scientific name, you always italicize, and it's always going to be made up of two parts. Scientific name is written with the genus followed by the species name. So whatever genus that living organism falls into, followed by the specific species name. When we write out the scientific name, the genus part is always capitalized, and the species portion is always lowercase. It is never capitalized. When we type it out, it is always italicized to keep it separate to indicate scientific name. If you were to write it out, sometimes it's hard to write italicized to distinguish from the rest of your handwriting. You can either write the scientific name and then underline it to distinguish that it is a scientific name, or you can write it in cursive. Here's an example of a bacteria written with its scientific name, Staphylococcus aureus. So as you can see, I have it typed out, so therefore it is italicized. The genus part is capitalized, while the species portion is not. When writing out scientific names, you can abbreviate by shortening the genus portion, so you can put G.species. That is the only accepted way to write out or shorten a scientific name. So for example, you can write out S. aureus. You can't write Staph aureus. That would be incorrect. So you're probably thinking, okay, well, what about viruses? We talked about bacteria, we talked about archaea, we talked about eukaryotic cells, but where exactly do viruses fall into this? Well, they're in a separate category. We call another category of agents acellular infectious agents. These are parasitic. That means that they must rely on a host, and the host will be damaged when they are used. Depending on which microbiologist or virologist you talk to, there are some who will argue that viruses or these acellular infectious agents are not truly alive, and therefore you can't actually call them an organism, you can't actually call them a microbe, yet they still often get classified in the groups as a microbe, and that's fine. I think there's still a lot that still needs to be understood about acellular infectious agents, but what's important to note about these is that they cannot survive on their own. Acellular infection agents that are left outside of a host cell cannot replicate. They cannot reproduce. A defining factor in biology that describes what life is, life means that you can reproduce. You can produce offspring. Acellular infectious agents do not have the mechanisms to do that on their own. Instead, they have to hijack host cells, use mechanisms within that host cell, and then can actually replicate within a host cell damaging the host. There are three main types: viruses, viroids, and prions. We will talk a lot about acellular infectious agents, especially viruses, when we discuss Chapter 6. So why do we care? Why do we care about microbiology? Why do so many people study it? Who cares, right? Well, microorganisms are extremely important. Well, first of all, we study them because some of them can make us sick, right? Some can be harmful. We call those pathogens. But what's even more important is that microorganisms are so beneficial to our survival. Not just our survival, but the survival of many other animals and other life forms on this planet. Microorganisms are extremely vital for the survival of life as we know it. Within your body carries at least 3 times more bacterial cells than human cells. You're probably thinking, okay, where's the Perel? Where's the hand sanitizer? I need that now. It's not the case. This is very helpful bacteria. You are full of helpful bacteria on your body and within you as well. This helpful bacteria we call normal microbiota or normal flora. It actually helps keep you very healthy. It prevents disease by competing with pathogens being the bad bacteria. They help teach your immune system how to respond properly. They help break down foods that your body may not normally be able to digest on their own. Not only are they helpful to you, but they're also helpful in the environment. They help with the recycling of nutrients. Some garbage hills actually will put certain microorganisms into the landfill to help degrade different compounds or different trash. A lot of microorganisms such as cyanobacteria produce oxygen. They produce a lot of oxygen, and if we were to eliminate these microorganisms, the oxygen level in our atmosphere would deplete so much that we would not be able to survive. Commercially, microorganisms have also been proved to be very beneficial. We use them in food production, baking bread. So yeast is a type of microbe. It's a eukaryotic single-cell organism. Also fermentation, making beer, yogurt, cheese, buttermilk. Biodegradation. This is actually becoming very popular right now with studies. There's something called bioremediation. We're finding that there are some microorganisms that can break down plastic. There are studies looking at taking these microorganisms and introducing them into the ocean in order to help decrease that level of plastic pollution. Bacteria or different microorganisms are used within biotechnology, genetic engineering. We'll talk a lot about that when we discuss genetics and biotechnology. Microbes are cheap, so we can use them as research tools. Yeast, being eukaryotic, just like you and I, provide a lot of information. So we can get a lot of information from studying a yeast that is actually applicable to us as well. And they're cheap. No one cares about yeast. People do care, however, if you started doing research on other humans or on elephants or on apes. Bacteria and yeast, no one cares. It's cheap, it's easy, and it's ethical. There are a lot of fields and occupations in microbiology. If you look at pages 4 and 5, there's a brief explanation of these different fields that's very useful to for many different industries. And being biased, being an immunologist and microbiologist by training, I think learning microbiology is very important no matter what your career path takes you. It's just good knowledge to have. If you have any questions whatsoever, feel free to email me. My email address is BARRK, the number 1, at matc.edu. Also, we will have some virtual classes this semester. They will be optional, but those are a great opportunity for you to check in if you do have any questions. You can come to those virtual classes, and then we can go into more details or into more I can go into more detail on any specific topics that you may have questions on. Okay, great, and have a great day.